Good morning, good day, good evening, everyone. And I'm very pleased to host and moderate today's webinar, which is titled Biologically Targeted Radiotherapy, Utilizing Imaging Biomarkers to Characterize Tumour Heterogeneity for Precision Radiation Therapy. Our esteemed speaker tonight is Professor Annette Haworth, who is the director of the Institute of Medical Physics at the University of Sydney, Australia. And she is also the course coordinator for the Medical Physics postgraduate program. She has more than 25 years experience as a clinical medical physicist, also working previously at the Peter MacCallum Cancer Centre in Melbourne. Annette's research interests have focused on novel approaches to brachytherapy and radiotherapy treatments, in particular, using quantitative imaging for biological optimization of treatment planning and treatment response. The webinar is being recorded, so the link will be also available within 24 hours. Following Annette's presentation, we will have a question and answer session, and please type your questions into the chat, and I will be then conveying questions to our speaker. You will also receive a certificate of attendance, and this seminar is credited 1 CPD or CME point. So without further talk, Annette, welcome to our webinar, and I'm handing over to you.
Thanks very much, Eva, and welcome everyone from so many places around the world. This is very exciting for me. I'm in Australia. It's 10 o'clock at night, and so I can imagine some people are up very early and all times through the day. So this is very exciting to have so many people on the line. I hope everyone can understand what I'm saying. Let me know if I'm speaking too quickly, too slowly, and I'll try and moderate the presentation. So I'll just what I'll do is what I'd like to do actually is start off by running a poll. So as Eva said, the title of my presentation is Biologically Targeted Radiotherapy. I'm going to be talking about quantitative imaging and tumour heterogeneity. So I thought it might be nice for me to know just what you know or what you think biologically targeted radiotherapy is before we get started. So Magdalena, could we run the poll now, please? Does everyone know how to do this? Oh, okay, yep. So just run through the questions. There's 5 questions, and just have a go at what you think is the most likely answer to each of these questions. Tick the box and then submit, and we'll have a look, and then at the very end we'll go back through these questions again, see whether you've changed your mind, and then I'll clarify anything that's perhaps not clear. So I'll just give you a moment to please answer these questions and submit, and we'll have a look at the answers. I can't see how many responses we've got, Magdalena, but perhaps we've got 637 people on the line. So perhaps when we've got a good number of responses in, we'll have a look at the answers. So how we're going there, Magdalena?
Okay, only 22 responses so far.
Okay.
And people still see the poll and answer the questions. You have to scroll down on the poll to get to all 5 questions before you can submit.
So don't overthink it, just have a go, and we'll have a quick look to see what everybody thinks. And shouldn't be too hard.
81 and growing.
Okay.
Can we have a few more responses? And these are single choice for each question. Is that correct, Annette?
Yes, just tick the one that you think is what you think is most suitable.
Okay, let's just give people another 15 seconds and then we close the poll. Okay, 200 responses. Okay, fantastic. Magdalena, can we close the poll? And are you able to show us the results? Okay, so this is the statistics.
All right, so we got the majority said data for the first one, what is in biologically targeted radiotherapy, the dose prescription is based on, and 43% said C. All right, I shall write that down.
Can we look at question 2?
Yep, question number 2. I can see which best describes the objectives of biologically targeted radiotherapy. And I think we've got
all of the above.
37% for F.
So now we have 307 responses.
Okay,
Number 3. An
okay,
imaging biomarker.
Maybe defined as, and we've got 43% said A. Okay. And for 4, tumour heterogeneity means, and we've got 55% for
E.
E. Okay. Interesting. Okay. All right. And the last one is biologically targeted radiotherapy. We've got the majority as B as 52%.
Yeah. Thank you. We can close.
B is 52%.
Excellent. Okay, we will repeat this test at the end of the lecture to test whether our knowledge and understanding has changed.
All right, let's get going with the presentation then. Good. Okay, so I hope you can all see my slides. I'm just going to change my window so that I can see the screen. Okay, so as I said, the title of my talk is Biologically Targeted Radiotherapy, Utilising Imaging Biomarkers to Characterise Tumour Heterogeneity for Precision Radiation Therapy. So Eva has already done the introductions. Yes, I am a medical physicist. I'm at the University of Sydney, where I'm the director of the Institute of Medical Physics, and I'm also the course coordinator. And like most academics, I do research, PhD supervision, and many other things as well. So a little bit of background. So yes, I am in Sydney in Australia tonight. So I'm actually at home, of course, like a lot of people, but the University of Sydney is on this in this lovely old building here. I've been here just over 5 years now. Before that, I worked at the Peter MacCallum Cancer Centre, which is Australia's only public hospital dedicated to cancer treatments and research. So I spent 10 years there as a clinical research medical physicist, specialising in radiotherapy, and I did my training at Sir Charles Gardner Hospital in Perth. So today's presentation, the learning objectives are to understand the basic principles of biologically targeted radiation therapy and how it's different to current standard approaches, understand the current limitations of the clinical application of biologically targeted radiation therapy, and develop a basic understanding of the principles of quantitative imaging and their role in staging, treating, and post-treatment surveillance. Now, for most of my talk, I'm going to concentrate on prostate cancer. This is where I've done most of my research, but I will talk about other cancers as we go, and certainly the principles that I will talk about are very relevant to many other solid tumours. So prostate, as many of you will know, is a gland in males. It's situated deep in the pelvis. The bladder sits above the prostate and the rectum sits below. So for external beam radiotherapy or radiation therapy, a patient lies on a couch and high energy X-rays are delivered with a linear accelerator focused at the prostate in this case. So the way we deliver the radiation is typically from multiple angles, all focused on the prostate in the centre here. So basically we have an anatomical definition of the target. In this case, it's the prostate. And we also, what we're trying to do here is bring our radiation beams in so that we're minimising the radiation dose to radiation sensitive organs, which include the rectum and bladder. But the point to mention here is that the radiation is delivered to the entire prostate. Using a linear accelerator is the most common way of treating prostate cancer with radiation, but we can also use brachytherapy. We can use high dose rate brachytherapy, where we use a single source that drives under robotic control through needles implanted into the prostate, or we can permanently implant radioactive seeds into the prostate. But at the end of the day, the goal is the same, typically is to deliver a uniform dose of radiation to the entire prostate. Now, over the years, there have been many advances in the way we treat radiation as a whole, but thinking of prostate cancer in particular, LINACs now are very high precision in the way they deliver the radiation. The machine rotates around the patient with an accuracy of less than 1 millimetre. We can utilise advanced imaging. So when we're doing treatment planning, we have advanced imaging capabilities to better define our tumour, better define where our organs at risk are, because we're trying to avoid them with our radiation beams. And during treatment, we can confirm the radiation beam is accurately focused on the tumour. So here is an example of MR compared to CT of the prostate, and we can see the prostate very well on MR compared to CT. And up here is an MR LINAC. So we're using an MR in this case to visualise where the prostate is before we deliver the radiation. Now, there's not too many centres in the world that have an MR LINAC at the moment, but this is becoming more and more common. We also have some advanced techniques. I won't go through all the details, but it's that basically we can move either the beams or the patient so that the beams are focused on the tumour the whole time. And in some cases, we can even vary the shape of the radiation field as the treatment progresses. And that's particularly so with the MR LINAC. So let's just talk about 3 facts about prostate cancer then. So these are Australian data, but they're typical of the sorts of data just scaled for the population, of course, around the world. So we've got about a population of around 25 million people in Australia. In 2018, almost 18,000 men were diagnosed with prostate cancer. Now, the 5-year tumour control rates, whether it be surgery or radiotherapy, are actually very good, in fact better than 95%. And this is compared to 59% in 1988, and this is a result of the advances in technology and imaging. However, men still die. So, and again in Australia in 2018, there were 3,500 deaths as a result of prostate cancer. So the data that I gave you a moment ago was 5-year data, but you know people are living much longer these days, and ultimately 30 to 50% of our patients will fail treatment, and it's a result of local resistance or systematic spread or systemic spread. Now, how can we increase long-term control rates? Well, we do know from studies that increasing the radiation dose that we deliver to the tumour can increase the long-term tumour control rates. But the more radiation we put into that prostate, the more chance we have of increasing the toxicity as a result of irradiating the surrounding healthy tissue. So the question is then, how can we increase the radiation dose to the prostate in this case, or the tumour, without increasing toxicity? So remember, we're bringing in our radiation beams from lots of different angles, all focused on the entire prostate at the centre of the pelvis in this case. So with prostate cancer, we do treat the entire prostate, even though the tumour itself may only occupy part of the gland. In general terms, that's because we don't know where the tumour is inside the prostate. That might be at the time of treatment planning. but also at the time of actually delivering the treatment, so we were being able to see the prostate, which is actually not that easy to see on CT anyway, but certainly not being able to see where the tumour is a major limitation. However, in recent times, we have some guidelines which tell us that we can actually use MRI. This is PIRAD's reporting system, tells us that ADC, which is from diffusion rated imaging, will give us some indication that a tumour might be present. But we'll have a look at some of the limitations of that in a moment. Now, the other thing to be aware of with prostate cancer, and it's the same with other tumours as well, such as breast cancer, is it can be multifocal disease. So we wouldn't want to just treat that larger lesion that we can see, because we'd be concerned that we're leaving secondary lesions behind, which could also be aggressive and later spread to other parts of the body. But however, there has been a suggestion that maybe what we should do is, rather than trying to increase the dose to the entire prostate, we should just see where the tumour is, where the largest volume of tumour is, and increase the dose to that region. So this was actually being done in the context of a phase 3 clinical trial called the FLAME study. So these results showed us that in actual fact, by boosting this tumour volume here that was visible on MRI, the 5-year biochemical disease-free survival rates were 92% versus 85%. So this seems to work. In terms of toxicity, well, there wasn't a statistically significant difference in the toxicity between the two arms for genitourinary and gastrointestinal toxicity, which is good news. But the thing is, these are still quite high rates. So we certainly need to be cautious that we don't really want to take the dose up any higher, because these would certainly be not good. But really we'd like to get these side effects down even further. So we've really still got two goals, which is often the case with radiotherapy. We're trying to increase the dose to increase tumour control, but we're also trying to reduce the dose to our organs at risk, because we want to do better. So in the FLAME study, their goal was to deliver a very high dose to the tumour. I think the standard dose was 78 gray to the entire prostate. I should have written that down, but the boost dose was 95 gray. So it was quite a big increase in dose that they were aiming for to the visible tumour volume. But in actual fact, what they found was, in most cases, patients did not actually receive 95 gray to the tumour volume, because if they had delivered 95 gray to this entire volume, it would have exceeded the dose constraints to the rectum or bladder and any other organs at risk, such as the urethra. So in most cases, they were not able to achieve the prescribed dose to the tumour that they had hoped. The other thing was that they did use MRI to define the subvolume of the tumour. But again, as you can see here, these images are quite fuzzy, and there isn't really a systematic way, well, there wasn't a systematic way of defining the tumour subvolumes in this study. This slide's perhaps a little bit out of place, but I just wanted to pause for a moment and say that these images here, of course it is a head and neck, it's not a prostate, but these are images using different parameters that you can get out of multiparametric MRI, so ADCs from diffusion weighted imaging. These parameters here, the KAP, VP, K-trans, are from DCE, dynamic contrast enhanced imaging. CT, I'm sure you all know what that is, and this is PET. Essentially what you end up with is different volumes, and that's basically because each of these parameters are representing different things. So this is something that we do need to be mindful of when we're looking at using different imaging sequences for delineating tumour volumes. Okay, returning to the FLAME study then. So as I said, there was a subjective delineation of the boost volume. So the next question that we might want to ask is, can we use an objective method for defining the boost volume? So in other words, we're all doing the same thing. So because we weren't able to achieve the dose that we were aiming for in the boost volume, The question is then, is perhaps there are radio-resistant subvolumes within the tumour. Perhaps we just need to take subvolumes rather than trying to take the whole tumour volume up to 95 gray. What if we were able to identify subvolumes that would actually benefit from this higher dose? Now, this is actually not a new concept. So the concept of biological target volume was proposed way back in 2000 by Clifton Ling. So way back then, Clifton Ling came up with the idea, and he was looking at MRI, he was looking at PET imaging, and he really just came up with the concept of, rather than just treating the entire tumour, which is the gross tumour volume in radiotherapy language, perhaps we could really just look at subvolumes, which might represent a certain characteristic of the tumour that might need a particular radiation dose. So this was a great concept, but back then in 2000, imaging studies were typically below resolution. So I'm talking here about MRI and PET. They were of poor quality. Imaging was expensive, it still is, and it wasn't widely available and certainly not validated. So it didn't get adopted very quickly. So there have been studies over the years. This study here is actually looking at hypoxia in the cervix. So hypoxia is when cells are devoid of oxygen. And what this group were able to do is they were able to find signals within multiparametric MRI. This happens to be a DCE, or dynamic contrast enhanced imaging, that correlated with hypoxia. When they took a group of patients who had been imaged, they found those with a low hypoxia score. In other words, the tumours did not contain significant volume of hypoxia. Then the progression-free survival was indeed superior to those with a high hypoxia score. So in other words, this demonstrated that hypoxia does lead to poorer outcomes. So in terms of the prostate, we again do have evidence that hypoxia impacts on biochemical failure. It's hypoxic volumes have the potential to metastasize, and they have a higher resistance to radiation therapy. However, it's not really well understood in prostate cancer. So some of the earlier studies were done with using probes, which doesn't tell you about the whole tumour volume, and these probes are not widely available in clinics. But this early work certainly demonstrated that there was something going on when you were able to define a tumour-contained hypoxia. But let's step back for a moment and let's just think of hypoxia as just being one of the parameters that could be related to radio resistance. Let's just take a purely hypothetical case here, which is a lung tumour. Now, for a physicist, let's say this lung tumour here has got brown cells, green cells, grey cells, and blue cells. And we give it some treatment, and what happens is the green cells are reduced in number, which is good. Brown cells are dramatically reduced in number. But look at the blue cells. They've actually continued to grow and metastasize. So retrospectively, this will tell us that the blue cells are the bad guys. So really what we would want to do, I've drawn the red circle around here, but this is during treatment, but really we should have drawn the circle around here and given a higher dose to these blue cells compared to a dose to the rest of the tumour. And that potentially could have overcome the issue of these tumour cells proliferating during treatment. So the goal of biologically targeted radiation therapy, and we call it BERT within our group, is that with biologically targeted radiation therapy, we can identify where high doses of radiation should be delivered, and we can work out how much radiation we actually need to deliver. So I'll come on to that in a moment. Now, this is, it looks like a map of Australia, but it's not. It's actually meant to be a map of a prostate. And over here we have a tumour which is bounded by a red line, and within it we have, let's say it's a hypoxic subregion or anything that could be radio resistant. So what we want to do is deliver a really high dose to this red region. We still want to deliver a high dose to the surrounding area. And of course, we still want to deliver a reasonable dose to the tumour as a whole. But this way, we have a better chance of being able to meet the dose constraints on the urethra, which runs through the centre of the prostate, the rectum, which is below, and the bladder, which is anterior and superior to the prostate. So when I say we can work out how much radiation to deliver, well, we could do that using the tumour control probability. Now, what we do know from previous experimental studies, that is, if you increase the radiation dose, and this is just an arbitrary graph here, if you increase the radiation dose, then the probability of tumour control increases. But of course, also as you increase the radiation dose, the probability of complications also increases. And so what we try and do, or what our philosophy is what we would really like to do is identify the plateau on the TCP curve. We're not trying to just increase the TCP indefinitely or just increase the radiation dose indefinitely. We're trying to find this region here where the TCP plateaus out, and that will tell us that's going to find us with minimum normal probability of complications. So here's a tumour control probability equation. We won't go through all of the parameters except to say that there are, of course, a number of parameters in there that will tell us how each voxel within this prostate will respond to a given dose of radiation. At this point, I'll say that we've already identified that previously we've had restrictions or limitations in imaging, and that was one of the reasons why biological optimization didn't take off. Another reason is because people argue that they don't know the value of the parameters to put into the tumour control probability equation. Our argument here is that if there's some way that we can use imaging to work out what those biological characteristics are, in other words, the value of the parameters in this equation, then we can customize the dose distribution for the individual patient. So can we do that? Can we take a map, imaging studies, divide it up on a voxel-wise basis, and from that extract information about the tumour? Well, yes, we can. So here we have K-trans from dynamic contrast enhanced imaging, which tells us something about angiogenesis. This is previous studies. This is not my work. R2 star from BOLD has been shown to be related to hypoxia, and ADC from diffusion rated imaging has been shown to be related to proliferation. And down here at the end, we have PET imaging. This happens to be PSMA PET imaging, which of course is related to metabolism. So quantitative imaging is not a new concept. We've been using CT numbers in radiotherapy for many years. That's probably one of the simplest forms. PET images, of course, we've been using SUVs, although this is really a semi-quantitative approach because it's all relative values. But now there is emerging evidence that multiparametric MRI, where we take multiple images and we fit mathematical models to these images to tell us something about the specific tumour characteristics. So for those of you not familiar with DCE, dynamic contrast enhanced imaging, for example, we can plot graphs. We image multiple time points after injecting a contrast. We can see how quickly the contrast is going into the tumour, washing into the tumour, how long it stays in the tumour, and how it washes out. And by fitting various models, we can work out various parameters and kinetics of the imaging. The role of PSMA PET in terms of localized prostate cancer is still experimental. It has been increasingly used in Australia to rule out distant metastases before a patient starts definitive radiotherapy. But in terms of biologically targeted radiotherapy, this is something that we're still trying to understand. So we did a study when I was at Peter MacCallum Cancer Centre. We did imaging on a whole bunch of patients that were scheduled for prostatectomy. We imaged the patients before surgery, and then we correlated the images that we saw on histology with the imaging studies. Now you can see here there's reasonably nice correlation. The pathologist has drawn around the tumour on histology, and we've co-registered the histology with the PET. So you can see we've got bright spots roughly within the tumour. But some cases that tells us absolutely nothing at all. This one tells us there's tumour over here, but it's missed this one. So the results are really quite variable. So what we do, rather than looking at each of the images, so I've just given you an example, that's probably an extreme example talking about PSMA PET, but the same goes for other MRI parameters as well. So essentially the idea we believe is to use predictive modelling or imaging biomarkers, if you like, where you take features, radiomic features. You can use machine learning or deep learning, whatever type of learning is the one of the day. Correlate these features in your imaging with ground truth. In our case, it was histology, because the prostates were removed from the patients, and then we can derive predictive models to tell us something about the tumour characteristics. And this is some work that my group has done, where we develop models so that we can predict tumour aggressiveness, well, anyway, Gleason grade, I should say, tumour cell density, and also hypoxia. So this is just again repeating what we did. So we had recruited 70 patients that were scheduled for surgery. We had histology, so this represents our ground truth. We had in vivo imaging, so this is multiparametric MRI and PSMA PET. We also used an intermediate step of ex vivo and a combination of deformable image registration and rigid registration, so that we could basically transpose the outline of the tumour that the pathologist had annotated directly onto the imaging studies, and then look for features within those tumour areas that told us something about the tumour. So these are our results. We've been able to predict tumour location. We're certainly not the only people in the world that have done this. We can also predict tumour cell densities. So the higher the density, again, the more radio-resistant the tumour is likely to be. And we can also predict tumour grade, or at least differentiate between high and low grade disease. So in terms of prostate cancer, I won't go into all the details. Most tumours have some sort of grading on the disease, but the higher the grade, the more likely the disease is to be aggressive. So in terms of hypoxia, hypoxia is a little bit of a challenge because you can't get ground truth directly from histology. So what we did radiogenomics, and this is where we again correlated our features from our imaging, but this time we took small samples from the biopsy specimen, and we did genetic profiling on those specimens, and we were looking for specific genetic signatures that have been shown to be related to hypoxia. And with that, we were able to identify 16 features within our imaging studies that indicated a relationship with hypoxia. So what I've talked about so far is biologically targeted radiation therapy. So a patient comes in, they undergo quantitative imaging, so this could be MRI, it could be PET, it could be CT. It doesn't work so well for prostates, but it does for other tumours. You combine this with clinical data. Of course, you never throw out the clinical data. You can include that, and then you can produce a treatment plan, and then you can treat the patient with brachytherapy or external beam or whatever your favourite radiation source is. Now, I'm not going to talk about treatment follow-up just a moment, just got a few more slides to show you on how this principle works. So this was some work done by a PhD student of mine. Her name was Emily Her, and this particular project is described in this paper. What she did was to demonstrate the biologically targeted approach. This is a proof of concept study. She took data from 5 patients. We had MRI data. We were able to determine the tumour cell density. So first of all, she produced a plan, this is plan A, using a conventional intensity modulated radiotherapy approach, 78 Gray to the entire prostate, which is a standard of care. She then produced a biologically optimized, and this was basically by modifying the radiation dose based on the TCP equation that I showed you before, using cell density distribution. And then she incorporated prostate motion. Now, the reason for that is essentially with using biologically targeted radiotherapy, you end up with a large number of small beamlets. So if you can imagine trying to treat this, but the prostate is moving, either because the patient's breathing or whatever patients might do in that particular area, then the treatment's not going to work. So you have to basically do robust modelling in the same way as you do with particle therapy. So this, she talks about this in her review paper, so I'll just leave that there for a moment. So it's in Cancers, published in April 2020, and Emily Herr was the lead author. And she talks about the comparison between using, still using quantitative imaging, but doing a dose prescription just based on the tumour volume, compared with the biologically optimized approach, where you're varying the radiation dose on a voxel-wise basis through the tumour, which is related to the specific characteristics, in other words, how radio-resistant each of the cells are contained within the voxels. So she, I won't go through this, but essentially she produced this IMRT plan, or the plan had already been produced, so this is a retrospective analysis. She calculated the tumour control probability and came up with a value of 0.61. So remember, when we're talking about tumour control probability, we're talking about relative values here. We're not saying it's going to be 61% for this patient. What we're saying is that another patient that was, let's say, 0.5, this one would have a better chance of tumour control than the one that's 0.5. She then produced a biologically optimized plan and was able to get the TCP up to 0.94. She then introduced what I'll call the wobble, accounting for movement, which we see on a day-to-day basis, and that dropped the TCP by coincidence down to 0.61. And then she re-optimized by accounting for the uncertainties in treatment position, and again was able to get the TCP value up to a very high number. More recently, Emily published this work incorporating hypoxia into the subvolume, again in Cancers. This was published last year. So I'll just go through this. What she basically did was she compared again a uniform dose of radiation to the entire prostate, just boosting the volume in the same way as they did in the FLAME study, and then boosting a subvolume region that was meant to be hypoxic, and doing the optimization based on the hypoxic subvolumes. Again, these results perhaps a little difficult to understand very quickly, but essentially this is when you, this is just two patients. When you deliver a uniform dose of radiation, if there's no hypoxia, you can get a good TCP. But as soon as you start introducing hypoxia into the equation, then the TCP drops very dramatically. If you now just boost the tumour volume in the same way that they did in FLAME, it certainly brings the TCP up. And for both patients that's shown there, but it's still a long way off really accounting for the hypoxia. But if you now account for the hypoxia, then you can really get the TCP up much higher. But this really does require very, very high doses of radiation to very small volumes within the tumour. So I'll move on now. Oh, so actually I was going to mention particle therapy. So again, I haven't really got time to go into particle therapy, and I'm not suggesting that particle therapy is the way to go for all prostate cancer patients, but in situations where potentially we couldn't really get those high radiation doses in with conventional radiotherapy, then particle therapy could be an option, maybe more for other radio-resistant tumours than for prostates. Okay, so now we'll look at using quantitative imaging for following patients up after they've had treatment. So this is some work from another PhD student of mine, Erin. So we've been running a couple of clinical trials where patients have been imaged before radiotherapy, and then we've imaged them after radiotherapy. This was the first one. We imaged them at 6, 12 and 18 months post-treatment. and another study where we have imaged at 2 time points before to look at variability or stability of features within the imaging, and then at 2 time points during treatment to see whether there is any potential for adaptive radiotherapy. Now, a big part of this project is actually understanding the stability of RMR scanners and the features that we get out of the images. So this is just to basically show the sort of things that what the images actually look like during the course of treatment. So a lot of work is involved in doing image registration, deformable image registration techniques, and doing mapping on a voxel-wise basis. And these results are not ready for public viewing yet, but I'll just talk a little bit about some of the difficulties that we've had in doing this. So Erin has published a couple of papers on this, looking at the repeatability, reproducibility and accuracy of features from quantitative imaging. So well, I'll just skip over that, but essentially she used a number of phantoms. She did the study over 3 different hospitals and measured at various time points. Again, I won't go through all the results of her study, but this is just a sample of some of the things that she found. So in terms of the accuracy, so she had a phantom with known T1 values and known ADC values, and so she had 3 different scanners, so she was able to compare the parameters, the ADC and the T1 that came out of the scanner with the known values within the phantoms. And you can see there's some quite large variations. In actual fact, accuracy is not something that we are worried about in our study, because we are really looking at the relative change on a patient-by-patient basis. But this tells you that if you were going to transfer your patients between scanners, that might be an issue. So we looked at differences for the individual scanners, and you can see the variation actually is quite good. And then between scanners, again, this is just looking at the relative values. It was just the R2 star that we saw quite a large variation, and we didn't have ground truth for R2 star, but just looking at the variation in R2 star values across scanners just gives us a little bit more variability. So this just gives us a feel for the potential of using quantitative imaging for tracking changes in response to radiation. So I'm getting to the end of the presentation now, but I didn't want you to go home thinking that we've only done this in prostates. We are interested, of course, in other cancers as well. So with liver cancer, there has certainly been some radiomic studies, and you can certainly see that there are various features in liver cancers. So we're actually looking again to see whether there is a potential for adaptive radiotherapy, particularly in high risk, I shouldn't say high risk, but in HCC liver cancers, where the liver has got a poor baseline function. So we're looking to see whether we can monitor the function of the healthy liver, or what's left of the liver that is healthy, during the course of treatment, to see whether we can dose escalate. In terms of breast, we're looking at multiparametric MRI and PET. So in this particular study, we're interested in breast cancer patients at the end of their chemoradiotherapy. They'll sometimes need to make a decision on whether to have a complete mastectomy or whether they can go with breast conserving surgery. So we'd like to be able to use imaging features to determine whether the patient really has had a complete response and can have conservative surgery. And another area that we are looking at is in colorectal cancer. So again, the situation here is in locally advanced colorectal cancer. The patient will typically have chemotherapy and radiation, and then they will go for surgery, and it's quite major surgery. But what they find is at the time of surgery, around 20% of patients have no evidence of disease. So in other words, if we'd have known in advance that the tumour had completely responded to chemorads, then they could have been spared that surgery. So this is all something that we're excited about in the future. So in summary then, I have shown you that imaging can define the spatial distribution of tumour heterogeneity. We can personalise the radiation dose based on this distribution of tumour heterogeneity. And we have the potential here to map the response to treatment. So in the case of prostate cancer, if we can see that there is a radio-resistant core, then potentially we could either boost that volume during treatment, so adapt the radiation biologically during the course of treatment, or if the patient relapses sometime after treatment, then we can work out whether we can go in and do a conservative, I should say a focal therapy. Currently, it's very difficult to know what to do with a patient that has recurred locally, whether you give them more radiation, just giving them a whole big dose of radiation to the prostate that's already been irradiated is not good. So this would help us be more precise in doing a top-up dose. But this is not standard of care by any means. So I've talked about some of the predictive models or the AI models. There's lots of models around, but they do need to be validated. We've seen that you can get variation in quantitative imaging. This depends on the mathematical models that you're using to fit your parameters. It depends on your scanning equipment as well. So our MRI scanners are not sold as quantitative machines. They're sold as imaging devices to show us pretty black and white pictures. We haven't quite got to the stage where they're recognised as quantitative machines yet. And then we also have to consider the uncertainties in treatment delivery as well. And I didn't list it here, but I did mention earlier that I think probably there has been scepticism in using TCP modelling or biological modelling in the past because of uncertainties in the parameters. But I'm hoping that over time that people will get an understanding and appreciation of what we can do with this approach. So that just leads me to say thank you to the many people that have been involved in the work that I've shown through this presentation. And I can now take some questions and then potentially we could have a look at the poll results again if we would like to repeat the poll.
Okay, do you want to do poll now?
Oh yeah, we could do the poll now if you like, Eva.
Yeah, wonderful. Thank you, Annette, very much. Can I please ask all the participants to redo the polls? Remember, you have to slide down the window to answer all 5 questions before you can submit your answers. Okay, Magdalena, can you tell us how the progress is going with answering the questions? Okay, we have 90 answers. Can I please have more people finishing the quiz? 150. And I have registered some questions. Once we finish the poll, the test, we will go to the questions and answers. 220, so maybe another 15 seconds. And then we look at the results. $300. Okay, Magdalena, would you be able to present the results?
Okay, that's great. So question number 1 then was in biologically targeted radiotherapy, the dose prescription is based on, and the majority there was C, data from MRI, PET and CT. So 74% had that answer. Yep, that's the one that I would have gone through with before we had 43% have that answer. So I'm pleased that we got that message over. Now, to be fair, you would actually want to use the clinical data as well. I would never say you just check out all the biopsy data and that sort of thing. But essentially, what I was really looking for was the imaging data for biologically targeted radiotherapy. It's giving us that spatial information. The biopsy report is only just, of course, taking one small sample of tissue. But if we're trying to basically do a dose painting by numbers, we're trying to vary the dose on a voxel-wise basis, we're going to need imaging to enable us to do that. The next question was, which best describes the objectives of biologically targeted radiotherapy? So before we had 37% said all of the above. Now we've got 56%, and yeah, that's pretty much the one that I would have gone with as well. So yeah, generally speaking, we are trying to both increase the tumour dose and reduce the dose to the organs at risk. I mean, obviously it will depend on the specific tumour, but the examples or the example of prostate that I gave with the FLAME study, for example, was that they weren't able to achieve the radiation or the prescribed dose to the tumour volume that they wanted in a lot of cases, and the toxicity was still reasonably high, and of course we'd like to see that much lower. So the longer our patients live, you know, the more important it is to keep an eye on that late toxicity. If we now go to question number 3, an imaging biomarker may be defined as, now we've got two high ones here. So the first one was objective measure of medical state, function or property, which can be measured. Before we had 43%, we've now got 46%, but we've also got 38% saying a type of image, e.g. CT, ultrasound, MRI, etc. And again, that's so the key word here was that a quantitative biomarker is actually not about the image, it's about the data that comes out of it. So really I was looking for A, which is the objective measure, rather than a type of imaging. But essentially, yes, it is imaging, but it's taking the numbers out of it rather than looking for dark spots or white spots. Yeah. Question number 4, tumour heterogeneity means, now the majority of people went with E, which is what I think we had before, was 55%, now we've got 63%. Yes, that's the one I was looking for. So a tumour has more than one type of tumour cell. So I showed you that lung case where we had blue cells and green cells. That's good enough for us physicists. But basically it tells us that each of those cells or clusters of cells will respond differently. So that's really what I was after as well. And finally, question number 5, oh, that's interesting. Biologically targeted radiotherapy is, and we've got 46 and 43% on B and C, a treatment delivered using the biological, biological agent and a radiation source, or C, a treatment that can be delivered using the standard linear accelerator. I was really after C, actually. So a biological agent, I mean, I suppose in theory, yes, it could be. It could be if you're using immunotherapy as well. But that wasn't really what I was referring to. That's sort of speculative and down the track. But I would accept both of those answers. All right, so that was great. That was interesting to see.
So now for some questions.
Yeah.
Annette, a question was raised about your 95 grade boost dose, which seems enormously high. What fractionation did you use, and after which dose did you start your boost?
Okay, well, that wasn't me. That was the FLAME study, and that was a Dutch study, and that's not a study that I was involved in. From memory, it was a standard, so I say standard 2 grade per fraction, that's the 78 grade that was delivered to the entire prostate. And so obviously just scale that up. So yes, so it was your standard 39 fractions was what they used in that particular study. So the Dutch, so that was the phase 3 trial that they ran. They have actually also done another study, which is called the HypoFlame study, where they used a hyperfractionated schedule, but that's a phase 2 study. And the results, again, look promising, and I didn't want to sort of go into that. It was just a little bit too complicated because I wasn't really talking about fractionation. But again, it looks like very promising that hyperfractionation actually might be the way to go with this particular approach.
Fantastic. Question about hypoxia. It is not a static feature. Could you please explain how do you account for changes in hypoxia during the treatment when optimizing the treatment for TCP?
Yeah, that's a very good question. This question always comes up because it has been shown, for example, in head and neck tumours that hypoxia can be transient and spatially unstable. But this is something that we are looking at in the studies that I showed to see whether we can identify stability in those features. So it's early days yet. It is an incredibly challenging study. We have collected the data and we're still in the early phases of processing the data at the moment. There are so many uncertainties that you need to take into account. Normalising the data and during a voxel-wise co-registration is quite challenging. So we've worked out what thresholds we want to use based on the uncertainties that I showed in the presentation. We've worked out how to do the co-registration, which is also challenging. So the next thing now is to see whether we can define. So remember, we haven't got ground truth on these studies. All we can say is what these features in the past have correlated with hypoxia. You know, how do these features stay stable? So the one I showed was the second study I showed is we're doing imaging at 4 time points, 2 before radiotherapy. So we'll be able to see whether it's stable without putting radiation in it. And then again, does it move after you've started the radiation treatment? I suspect we won't find much in prostate. It's probably more useful in other tumours such as head and neck, but nobody knows the answer to that question yet. I'll tell you when I know.
Now, how was the clonogenic cell density used for TCP calculations retrieved from MR images?
So in the TCP equation, of course, you have got a number of clonogens. So the way we did it, it's a little bit complicated, but when you look down a microscope, of course, what you see are nuclei, and we can count nuclei. There's ways of counting nuclei. That's relatively easy to do, but that, of course, counts everything, both your normal cells as well as your tumour cells. So what we actually went through was a normalisation process, and we used published data to show what is the normal percentage. So we knew what the Gleason grade was, and we knew what to expect. So we were able to basically normalise the data so it matched published data before to come up with our distribution. So that was the unique part of it, was producing a distribution of cell density in 3 dimensions to apply the voxel-wise optimization.
Excellent. Now, from your work, do you plan to publish guidance on scanner performance metrics that must be QA'd for effective quantitative follow-up imaging in radiation oncology?
Yeah, so that was the part of the purpose of the QA study was to work out just how often you actually do need to do QA. I mean, what you really want to know is, do I have to do QA every day? Can I just do it every 6 months, every 12 months, you know? And then obviously the next issue is if I'm going to have patients on different scanners, which we didn't, you know, what would you need to do more QA? So as you could see by those results, we did find that the scanners were reasonably stable. You know, it's like most things, it's always makes you feel good when you actually do QA on the day, just as we do stereotactic treatments, for example, cranial work. We will do QA on the day just to make sure that we couldn't believe that Linac could suddenly go out overnight, but you just don't want that to happen. So, you know, I think this is something that we'll all get a better feel for over time. There's a lot of people doing work in this particular area, But at this point in time, we feel confident that the 3 scanners that we used, and they were all from the same manufacturer, they were all using the same protocol. We've applied the same mathematical modelling to produce the parametric and pharmacokinetic maps. So we've reduced all of those uncertainties and variations. And so really we're just left with the variation in the patient, which is what we want to measure. And the variation in the scanner itself is really quite small, as you could see from the data that I showed.
Can you suggest the best phantom to use for MRI QA for these purposes?
Well, that's a very good question. We used the NIST phantoms because they were available. So if you just do a search on the CUBA group, the Quantitative Imaging Biomarker group, they do talk about various phantoms. So the NIST phantoms are the ones that we use. That's the commercial phantoms that we used. We did actually also produce our own sort of small phantoms with different inserts. Now that was really just looking at the stability, because we essentially had one phantom that we passed around all 3 scanners because we wanted to, obviously compare the scanners. So we had one commercial phantom that we passed around. Now these phantoms have a life of 3 years, and that was how long that study was. So obviously these things are quite expensive, so it is quite useful to have a network where you can sort of take it in turns to buy a new phantom. So we then just produced our small in-house phantom with basically test tubes of different concentrations of materials in them, and then we replicated those at each centre, and that was basically just testing for reproducibility.
Fantastic. Now, when doing voxel-wise response analysis, does the tumor deformable image registration become a big concern? How do you evaluate the potential impact of the registration to translate it to the response quantification?
Yeah, that's a very good question. It is very, very challenging. We're in radiotherapy. We are very used to going through, bowel preparation, for example. The imaging people perhaps are not quite so used to doing that as we are in the radiotherapy world. And certainly if there is gas in the rectum, you get these artifacts, and it makes it very, very difficult to do the co-registration in the first instance. But then the question is, of course, how reliable are the voxels around the artifacts? So certainly the things that we have learned is, bowel preparation, bladder preparation, and even bladder preparation is complicated because the patients are on these scanners for around 40 to 50 minutes, and obviously you're going to get bladder filling happening in that time. So it is quite demanding, and it's very difficult to do. There's no doubt about that. But the way we manage it, basically, essentially what we have done is we obviously normalise your voxel size to a certain extent. When we're creating our predictive models or looking at features, if you like, we apply what I call a donut of uncertainty. So you're looking at where you think the region is. You then apply a region around it where you say, well, I'm not sure what's going on there because of variations or other uncertainties. So that's for the predictive models. But in terms of this voxel-wise co-registration, yeah, it's very difficult. I mean, we're actually looking at a combination of region of interest analysis as well as doing voxel-wise analysis. So with a region of interest, of course, you've then got more voxels, so you haven't got the noise issues. But really what you're then looking at is really your changes in the uniformity. So, you might start off with a huge amount of heterogeneity in the values through your tumour. And then as you deliver your treatment, of course, you're hoping that then that normalises out. So that's what you do when you're doing the region of interest, and that's the easiest way of doing it. If you're doing it voxel-wise, then yes, it is absolutely really difficult to do. You've got much larger uncertainties, error bars, if you like. So determining what is a true response and what is a response, which because you just pick the wrong voxel, there's an error, does make it very difficult. So you're really looking for those massive changes. And of course, you can use clusters. I mean, you really, at the end of the day, go, oh, I've got a whole bunch of voxels here that, look like they've changed. You kind of draw a circle around them, and that's a sort of a region of interest within the region of interest.
Annette, I will finish with one last question. When do you see biological treatment optimization be implemented in clinics? and what steps need to be done to implement it?
It was interesting. Someone else, I was at another presentation, someone else was asked that question and they said 5 years. And, you know, look, yeah, I think probably, I don't know whether it's 5 years or whether it's longer. The way I answer that question is, I think with anything that we do in radiotherapy, it becomes stepwise. So what are we doing at the moment? We are taking a blob, which we see on our black, white, grey images, and we draw a circle around it. Well, surely we can do better than that, right? And so, yes, we can do better than that. We can certainly start using AI. We can certainly start doing retrospective. But we all want to boost those subvolumes. We've been doing that intuitively for a long time. Now we're starting to do it a little bit more systematically because of flame, but we've still got this issue of flame being a problem. So I think that we'll start off by doing it more in a qualitative approach and then in a quantitative approach to actually achieve what it is that we want to do. But I think we're a hell of a lot closer to doing this than we were, say, when Clifton Ling first suggested this 22 years ago. Yeah. So yeah, I think it's going to be a stepwise approach for all sorts of reasons: the validation, the collaboration, the evidence, but also just the access to the imaging as well.
Great, fantastic. Annette, thank you so much for a wonderful, inspiring talk, and thank you for spending time with nearly 900 physicists in the middle of the night in Sydney. And before I say goodbye to our participants, allow me to share screen one more time. I would like just to announce that we will have another webinar. It will be held on 20th of April at 12 p.m. GMT. That means 1 hour later than today. And it will be on radiation doses of 100 milligrays. Do they represent significant risks? And we will have an exciting panel of speakers, and I am inviting all today's participants to join us again in April. So can I please ask everyone to join me in thanking Annette for excellent talk, and it's good night, goodbye, good day, good morning, and talk to you next time. All the best, bye.
Thank you very much. Thank you for inviting me. Bye, everybody.
Our pleasure. All the best.